Assalamualaikum warahmatullahi wabarakatuh. Hello, my name is Bagas Putra Ibowo from 12th grade Science 1. On this occasion, I will discuss and test foods for carbohydrates and protein. Before I test it, we must first know that our bodies require a small amount of carbohydrates and protein to perform daily activities. This food can be obtained from foods consumed daily. The foods we consume on a daily basis are carbohydrates and protein. Well, some tests of this food are used to test the ingredients. To be clear, let's do a test on a food item for the concentration of carbohydrates and protein. Let's do it.
The carbohydrates and proteins tested this time are bird eggs, from which we separate the egg whites and the yolks, then the bird potatoes, then the bananas, rice, and toast. A useful instrument for testing foodstuffs was a beaker glass to hold a solution of copper to sulfate, sodium hydroxide to test proteins, and aqueous, and then betadine to test carbohydrates, mortar and LUF for use in fuel, 3 drip plates to sample refined ingredients, a 2-dropping pipette to extract solution, and a spatula to transport the items from the mortar to the drip plates.
Okay, once we see the necessary tools and materials, now we can start the test. First thing we do is label on the drip plate. Here I have written egg whites, egg yolks, bananas, bread, potatoes, and rice. The label function on the drip plate is to facilitate the identification of the finished material, which allows you to distinguish between the beginning color and the end color. In here I prepared 3 drip plates. The first one I wrote early, a food item that didn't read any solution as beginning color. Then the second one I wrote was a protein test that would be treated with copper sulfate and sodium hydroxide indicator to test whether there was any protein in the food. The third I wrote down a carbohydrate test for later dedication in betadine to test for carbohydrates on foodstuffs.
Protein and carbohydrate tests are tests that use discoloration identification. Now we start for the groceries. The first one, we flatten the egg yolk.
In the process of preserving, we can provide the aqueous to facilitate the healthy and the secretion of food series, so that it is easier to identify protein or not. For the aqueous, depends on the initial texture and the amount of food used. For example, egg yolks need more because the texture is dry, unlike bananas that have a content of water. It may take a small amount of aqueous.
The ingredients were mashed until the texture resembled a porridge. Move the delicate egg yolk to each drip plate.
After all the material has been smoothed and has been transferred to 3 drip plates and has been laid in this line, the center drop plate is the beginning and will be compression. Substituting the right drop plate for the carbohydrate test and the left drop on the protein test. Now we're going to drip each of these white indicator solution that indicates whether or not there is protein or carbohydrate content.
The first, we will test protein using solution sodium hydroxide and copper 2 sulfate. In here, I place the solution in an empty basin in order to distinguish the initial solution from the existing solution with the food. Don't forget to use the same dropping pipette. To test the proteins, first apply it to a solution of sodium hydroxide. Wait a minute. After waiting for a while, it was given a solution of copper 2 sulfate. Sodium hydroxide and copper 2 sulfate reacted to the peptide proteins bound on the sample. The presence of proteins showed the sample change into purple. Color formation is due to a complex of Cu+ ions with proteins peptide bonds. The second, we will test carbohydrates using betadine. In here, I also placed the betadine in the empty basin to distinguish the early betadine from those who had the error direction by the sample. Now, we're going to drip each 2 drops to the sample. The samples decoded by betadine changed color to dark blue, a result of a bond between the iodine in betadine and the amylin in the foodstuffs from a dark blue complex. The egg yolk and egg whites indicate discoloration to purple, which means it has proteins, while bananas, bread, potatoes, and rice indicate no discoloration, meaning no protein. Previously, I wanted to use the tempeh and tofu as a high protein sample, but the growing price of soybean made the craftsman strike off production. The egg yolk and egg whites indicate no discoloration, meaning no carbohydrates. The bananas indicate discoloration to dark brown, which means having low carbohydrates. And on rice, potatoes, and bread indicate discoloration to dark blue, means having carbohydrates.